go.
Well, thank you very much. Nice to be here. So there's a lot of different sustained release technologies out there. We heard some today, there's some others. One of the things that Ripple is unique is that there's no polymer involved. So it's only this prodrug technology we call Epidel. And the advantage is surface-mediated drug release. There's no polymer residue or implant swelling. And when the drug is gone, the implant is gone. So for physicians, if you see the implant, the drug is active. And because there's no polymer, you can make smaller implants, high drug loading. We have these nice zero-release kinetics, and it's very tunable based on dose and duration, either by chemistry, changing the underlying linker technology, or by surface area. And from a patient point of view, the idea is to be in the therapeutic window longer without the spike release that has the deleterious effects of some of these burst releases, to really improve long-term outcomes with a reduced treatment burden. And from a commercial point of view, we've been able to get composition of matter patents, even though many of the drugs we work with are off patent. And despite the fact there's no polymer, we can use standard polymer processing techniques, which leads to very scalable manufacturing. The other thing we have that a lot of the early stage drug delivery companies don't have is evidence of clinical translation. So this is our dexamethasone intravitreal implant we call IB814. You can see that the PK and a really nice translation in the clinic, in this case with our DME patients, where we saw a significant improvement in vision. From a strategy point of view, we have a balance of proprietary programs and partner programs. So on the partner side, we've done licensing option deals with AbbVie for, we call RDC 620, which is a bimatoprost intracameral implant. With Bausch & Lomb is focused on a retina program, and with Glaukos, they haven't disclosed where that program is. And then we also have the aforementioned IB814, where we have active negotiations right now on the partner side. And we have a number of partners that we've been speaking to that are interested in looking at using our technology with their drugs. But on the development side of it, we're working with a program on neuroprotection that we won't talk about today. And I want to focus today on the retina side and our two focus areas for dry AMD and GA, a CFB inhibitor and statins. So everybody in this room knows it's a big market opportunity with dry AMD, and the two products that are on the market have done really well. And both of them have managed to have really nice exits, and congratulations to Apellis with the deal that they did a few weeks ago. We think there's an opportunity to have a more effective approach to both geographic atrophy and intermediate dry AMD, one that is not only more efficacious but also safer, and that reduces the treatment burden. And we think in order to really effectively treat not only intermediate dry AMD but even geographic atrophy, you need to have a 6-month duration. So our focus is twofold. The first is a complement factor B inhibitor. The main advantage of complement factor B is its specificity, up with the change from the C3 and C5. And we think local drug delivery is the key. So from a data point of view, we've been able to establish first these really nice zero-release kinetics, the duration, safety in terms of tolerability, and we did an early PK study to make sure that we were above the EC50 and IC50 range. Today we're announcing the work that we've done with a laser CNV model, which we saw a significant reduction in lesion area, as well as complement inhibition, really much more significant than some of the products that are on the market. And the second area of focus is intravitreal statins, which have the potential to impact multiple aspects of dry AMD. And just in the interest of time, just quickly to jump to the data, we've been able to show, again, nice surface erosion drug release, and we've done NHP tolerability studies with several different doses to make sure that it's safe. And we now have, in a rabbit model, been able to show, despite the fact that these are normal rabbits, both precursor and clearance impacts on biosynthesis, and in the laser CNV model, significant reduction in lesion area and macrophages. And we have more data coming out in the next 3 to 6 months from an NHP model, which I think will be very interesting. So our strategy is really to raise a Series B. We did a Series A back in 2020, 15 million raised, but we've raised 65 million from partner programs or non-dilutive financing. We'd like to raise 15 million to get one of the dry AMD programs through IND-enabling CMC and GLP tox and early clinical data, phase 1, 2A. If we're not able to raise that money, we're open to partnering, so we're talking to to people on both fronts. If we're able to raise more money, we'd like to actually take the statin program through as well. But we look at the CFB inhibitor as being a best-in-class, and the dry AMD, statins for dry AMD as a first-in-class. Thank you very much.